Hi, everyone. I'm Steven Becker, the Associate Director of NEI's Office of Regenerative Medicine, and I'd like to welcome you today to the National Eye Institute's and New York Stem Cell Foundation Research Institute's joint webinar on a new induced pluripotent stem cell repository for age-related macular degeneration, or AMD. The National Eye Institute has launched the AMD Integrative Biology Initiative to support research into understanding the initiation and pathology of disease. Over 60 patient-derived induced pluripotent stem cell lines are now available to the vision community. These stem cell lines were derived from participants in the age-related eye disease study known as AREDS, or AREDS-2, the subsequent study. And these initial cell lines have single nucleotide polymorphisms that are known to confer high risk towards developing age-related macular degeneration. Because the AREDS-2 cohort has extensive clinical and medical data available, the hope is that phenotypic correlations with ocular cell types derived from these lines will spur better understanding of how age-related macular degeneration starts and progresses. These lines are now available at the New York Stem Cell Foundation's Research Institute, and you will hear more about how to obtain these cells during the webinar. Today, we'll hear from three panelists. The first is Dr. Kapil Bharti. He is a senior investigator at the National Eye Institute that leads the unit on ocular stem cells and translational research. He is currently leading a clinical trial with autologous stem cell-derived retinal pigment epithelial cells. Dr. Bharti will discuss the value of patient-derived induced pluripotent stem cells for AMD research and therapeutic discovery. Dr. Bharti, please.
Thank you, Steve. So, hi, everyone. I'll just quickly give you a very high-level overview of this whole project, how it started, and what's the value of using patient-derived iPS cells for disease modeling, particularly for age-related macular degeneration, or AMD. Most of us who work in this field know this part very well, that AMD disease processes extend across the RP-choroid interface all the way from photoreceptors to the choroid, I would say. This slide illustrates kind of a strategic way how RP is positioned between the photoreceptors and the choroid blood supply, and on its apical side, it's maintaining health and integrity of photoreceptors, and on the basal side, it's maintaining the health of the choroid throughout its life. And what happens in advanced disease stages of AMD is that RP atrophy leads to both of these stages called the dry stage or the wet stage. On the left side of your screen, you're going to see how disease is happening in case of a dry AMD schematic showing that disease initiates with sub-RPE deposits that are present under RPE cells in between RP and the choroid. And as the disease progresses, it is seen that there's RP atrophy, even RP cell loss, which leads to photoreceptor cell death eventually, and leading to really areas where patients are blind in the part of their eye. And that's how the disease also keeps spreading. As RP cells continue to die, the photoreceptors continue to die, preceding RP cell death. The other case of advanced stage AMD called wet AMD, and it's called wet AMD because the choriocapillaries are known to overgrow in this condition, and they kind of, what I like to say, go in the wrong Z direction, where they penetrate through the RPE barrier and leak blood and fluid into the subretinal space, again separating the photoreceptors from the RPE and causing their death. Needless to say, in both these advanced stages, patients go severely blind. So many different studies have done a lot of analysis on what are the causative factors behind AMD. And here's kind of a very simple schematic of what all of these results, all of these publications have led to. Epidemiological studies have shown that high-fat diet, smoking, and aging are the three major risk factors, environmental factors for aging. Whereas GWAS studies have linked many pathways that have been linked to AMD progression, including complement, lipid pathway, oxidative, metabolic, and ECM pathways. And it is thought that it is a combination of the environmental factors and the genetic factors that lead to this chronic stress around the photoreceptor RP choroid interface, leading to inflammation, perhaps abnormal complement activation, increased oxidative stress, metabolic changes in and around the RP, that of course lead to dysfunction of RP cells, leading to the sub-RPE drusen deposits that I showed in the previous slide. And eventually, with time, as the deposits build up, RP cells continue to dysfunction, they die off or atrophy, And that leads to these advanced stages, either the dry stage or the wet stage of AMD. So if one scans the literature for AMD disease models, over the past 50 so years or 55 years, a number of papers, over 3,000 papers have been published. Actually, most of them have been considered in the last 25 years that have used various disease models to model AMD, including rodent models and other animal models, plus cell lines in vitro cases. For instance, ARP19 is one of the most well-known RP cell line. Now, a few things that are missing in these models is that most of them lack connection to human genetics. I showed you that many papers have shown through GWAS studies that AMD is indeed a genetic disease. It is a vast portion of it can be explained by polygenic risk alleles. And then the other part of this is that AMD is a disease of the central part of the eye, which is the macula. But in most of the rodent models or in vitro cases, for instance, ARP19, there's no possibility of analyzing macular cells versus peripheral cells, or center of the eye versus a periphery of the eye. And obviously in animal models, you can't really do high content imaging or high throughput screens or biochemistry at that level. And lastly, to say is that ARP19, as popular as it is, it still often lacks a fully polarized RP phenotype. So with that, I want to introduce iPS cells, especially patient-specific iPS cells, as a new model to study AMD in vitro. And the beauty of patient iPS cells is that, first of all, it's patient cells, right? So you start with patient's genetics. So if you start with different levels of risk alleles, which we will show you in today's webinar, then you have that possibility of comparing different risk alleles to the lines, to the iPS cell lines that do not have risk alleles. So the way you can do it is you start with the patient's somatic tissue, reprogram those cells into iPS cells, and you can make literally any cell type of the eye or the body, if you have interest broad in immune system and its role in AMD, from those iPS cells and study their interaction with the eye cells. Here I have listed 3 examples of interest to us: differentiated RPE cells, or a 3D differentiated RP choroid, or photoreceptor cells, and you can make combinations of those. And in many cases, or in all cases, you can use them to model disease using physiological stressors. Again, the ones that we listed, including complement, metabolism, oxidative stress. All of those stresses can be used with these in vitro models made from iPS cells. And that gives you the possibility of studying disease at a very early stage, from initiation all the way to progression, and determine or discover treatments that can intervene at very much earlier stages and hopefully bring those treatments back to patients. So just in the interest of time, I'll keep this simple to just one model today, which is pure polarized RP monolayers made from patient iPS cells. And the way this can be done, again, there are many protocols that have been published to make RP cells from iPS cells. This is our favorite protocol that we developed in my group, where we can go from iPS cells to mature RP in about 10 weeks. The beauty of this is that we like to call it guided differentiation, or developmentally guided differentiation protocol, where we coax the cells into various developmental stages from iPS cells all the way to mature RP. For instance, they will go to neuroectoderm, RP progenitors, committed RP. And this is done by using developmental factors that have been used, that have been discovered for decades in various systems, how RP cells are made. Now, needless to say that at the end, we get fully mature and polarized RP cells. I'll show you examples of that in a minute, but here you see a pure culture of RP cell, and you can see they're fully homogeneously pigmented. And in this panel, in the flow panel, you can see that we've looked at it by many different markers. These cells, we can generate more than 98, 99% pure. And also here's an example that these cells have a very homogeneous phenotype. At least with this marker, we can tell that they have a very homogeneous phenotype. Now, if you were to analyze a disease like AMD, where we know it's highly heterogeneous, there are many risk alleles involved, that means we need to be able to make RP from every single iPS cell line we analyze. And this protocol at least gives us that possibility. And I can show you, or I can provide data here, is that this protocol is highly reproducible. So far in our hands, we have done it over 50 different donors with different iPS cell lines, more than 100 clones total. And we have done more than 500 differentiation runs, and every single time more than 95, 98% success, we have been able to make mature RP from these iPS cells. Here are examples of some of the iPS cell lines we have used, both research grade, clinical grade, healthy cells, AMD patient cells, different diseases. In all cases, we have been able to make RP from this using this protocol. And I think the biggest validation of this is that by now we have shared this protocol with over 24 labs, and they have been able to reproduce this protocol very well in their hands. And I think that's really important because it's not only working in our hands, it's working in the hands of other investigators as well. Here's some of our publications listed where we have published this protocol and the cells that have come out of this protocol. So now it's important that we understand that the cells, RP cells made using iPS cells, what is their phenotype, how functional they are, how validated they are, that how close they are to native RP cells, that can they really reproduce a disease phenotype, right? So I'm going to show you a couple of examples that we are highly convinced that the cells made with this protocol do lead to fully polarized and functional RPE cells. For instance, on this slide, you're seeing, first of all, RP cells here, they're stained with two different markers, 01 that stains RP junctions in green, and beta-catenin, that is a transcription factor that regulates canonical Wnt signaling, is in red. And normally beta-catenin during RP development is in the nucleus. As the cells fully differentiate, it moves out of the nucleus and lines itself around the cell borders inside the cell. And that is one example, one phenotype of full maturity of RP cells. And you can see here beautifully, beta-catenin lines itself around the cell borders, suggesting that these cells are indeed fully maturing, which is going to be important when you're analyzing an AMD-like disease, which is a late onset disease. You want your cells to be fully mature, not fetal-like, not undifferentiated. There are other ways we can prove that. In this example, I'm showing you two different images. On the left is a transmission EM image. So we're looking at the cells from a side view. On the right is a scanning EM where we're looking at cells from a top view. And I hope you can appreciate in the transmission EM here, the fully polarized nature of RPE cells. Here you have a nucleus that is basally located, melanosome that are apically located, and a confluent apical processes, which you can also see on this side here on the right side using the scanning EM. So many examples, and here are tight junctions between neighboring cells. So many ways we have demonstrated that the cells made with this protocol and from iPS cells can be made into fully polarized RPE cells. Now, structure is one thing, but most important is the function, right? So here's an example to show you that RP cells made from iPS cells do have physiological stimuli that are very similar to native RP cells. So in this graph, we're measuring multiple things. Let me walk you through it. So first of all, on the right side, we're measuring the transepithelial resistance of RP cells. People those who work in this field know that when two RP cells come together, they form tight junctions. As a monolayer forms, the tight junctions between neighboring cells become so tight that when you put an electrode on top and bottom, you pass current, cells will show resistance to the flow of current. And that we have measured is that we have demonstrated is that resistance is often directly proportional to how mature the cells are. At the same time, as the tight junctions form and the cells are becoming mature and there's transepithelial resistance, cells have transepithelial potential. What that means is that the apical membrane has a different resting potential than the basal membrane because both of them have now carry different channels on each side because the tight junctions don't allow a free flow of channels from one side to the other. And we can measure that here as the 1.5 millivolt, almost similar to native RPE cells. Excuse me. And then we can do physiological perturbation like reducing the potassium concentration from 5 to 1 millimolar, which happens every time light hits the photoreceptors. As a result, RPs and the potassium concentration in the subretinal space drops. RP cells open up their potassium channels, hyperpolarize to correct the potassium concentration. At the same time, ATP secreted by photoreceptors causes a classical depolarization pulse in RP cells because it activates through purinergic receptors, intracellular calcium stores that drive the basolaterally located chloride channels. So first of all, the response is very similar to native RP cells. And second, it tells you so many different things are right in these cells. Their junctions are right, their channels are in the right places, their purinergic receptor is working, the calcium signaling is working, their chloride signaling is working. So this is a very important assay that tells us that the cells RP cells made from iPS cells are fully validated to be functional RP cells. Another important thing I do want to say is that these cells are stable. So we have kept iPS-derived RP cells for months, 6 months, and have measured these electrophysiological responses 6 months apart from the same culture and got identical readings. So really, this is very important that we can do long-term analysis of these cells. If somebody has an assay that goes, it's a chronic assay, they want to go for a long period, the cells themselves are stable. Cells made from iPS cells, RP cells made from iPS cells are highly stable. And then the beauty of this is that iPS to RP differentiation is developmentally guided, right? So that means we can developmentally make them either macula type or far periphery type. I'm not going to go into too much details, but people, those who work in the RP field, know that in the macula, RP cells are slightly different than they are in the periphery. Here on the scanning EM, you see an example that the cells in the macula have these paddle-like apical processes that wrap around the cone outer segments, whereas in the far periphery, have finger-like apical processes that interdigitate between the outer segments. So the beauty of iPS cell technology is that it allows you to make macula type RPE versus periphery type RPE. And now, since AMD, which is a disease predominantly of the macula, one can study from the same patient cells. Do the cells behave different? Do the macula cells show more disease phenotype in a dish than the peripheral cells? And if they do, what are the gene signatures behind that? What are the pathways behind that? And that will be the best internal control we will have to say why disease only happens in the center of the eye. So with that, I just want to quickly show you a couple of phenotypes, AMD phenotypes that can be created in vitro using these cells. On top, you see an example of, so all of these are RP cultures, iPS RP cultures. On the left is control RP. On the right, we call AMD, is that cells have been treated with a physiological stimulus that leads to AMD-like phenotype in the cells. For instance, in this case, sub-RP deposits. You can see notice a big deposit underneath RP cells. It's a section of RP. You're looking at from the side. This deposit is stained with a H&E, so hematoxylin and eosine stains for these deposits here. On the bottom, you see another example, again, a side view, but fluorescent staining. In green are the tight junction markers, the actin staining that stains the cell borders and the top of the cells. And in red, we're looking for lipid deposits. And you can see again that AMD stimulus is able to stimulate formation of both sub-RPE and intracellular lipid deposits in RP cells. So these cells are able to replicate several of the AMD phenotypes in vitro. So with that, I just want to show you my last slide, and my colleagues will follow up on that. We have the first set of lines that were made were the highest risk allele containing lines. That's aligned with 11, a total 11 lines. 6 of them that you see at the bottom of this table have the highest risk alleles in the CFH gene or locus and in the ARMS2 HTRA locus and other complement pathway loci. Whereas the other 5, they lack the ARMS2 HTRA risk alleles on this locus, but they contain the other 2. So they provide a perfect control if somebody was interested in the role of ARMS2 HTRA loci as opposed to CFH or other complement pathways. So all of these are available, and my colleagues from NYSCF will show you the additional lines that they have made in collaboration with NEI that are currently available. And then another colleague, Kerry Goetz, is going to show you a database that has been developed by NEI that is now housing the medical history, genetics, and hopefully in future cell biology of cells derived from these iPS cells, with the goal to make a connection between all 3 using perhaps artificial intelligence. The last point I do want to make is that at least for this 11 sets, my lab has validated that all of these iPS cell lines do make polarized RP monolayers. So they're functional in terms of their ability to make RPE cells. With that, I want to thank the people in my lab, especially for the protocol optimization, Ruchi Sharma, late Janine Davis, Jin Won, Josh Miyagishima, David Ertolon, and Arvidas, and for disease modeling of AMD, Ruchi Sharma, Malika Nimagara, David Ertolon, Devika Bose, and Robert Ticini. Thank you very much for your attention. Back to you, Steve.
Thanks, Kapil. That was great. If anybody has questions, please use the Q&A function at the bottom, and we'll do our best to address them either during the presentations or we've reserved plenty of time afterwards. Next, we have Laurenbauer, who is Associate Vice President of Scientific Operations and Project Management at the New York Stem Cell Foundation Research Institute. She has been there, She has been there for about 11 and a half years and oversees the lab operations and project management teams, as well as the stem cell derivation group on the global stem cell array. Lauren will present information about the collection of AMD iPSC lines now available to the research community, how they were generated, and the rigorous quality controls used. Lauren, please take it away.
Thank you so much, Steve. It's a pleasure to be here to talk about this resource that's now available to the AMD research community. I did want to just introduce the New York Stem Cell Foundation Research Institute for those of you that might not be familiar. We are a nonprofit research organization, and our mission is to accelerate cures for the major diseases of our time through stem cell research. We do that in three main programs. Our NYSCF Innovators, where we have grant awards for postdoctoral fellows and early career investigators in labs around the world. We have our NYSCF Outreach, which is our events and our education initiatives. I do want to point out that we have our biggest event, the NYSCF Conference, which is a translational stem cell research conference held each fall. So I encourage you all to save the date for October 19th to 20th of this fall. And then we also have our NYSCF Research Institute. So we have over 75 scientists and engineers here on site in our labs. We have disease-focused teams in neurodegeneration, diabetes, women's reproductive cancer, and more. We also have our NYSCF Global Stem Cell Array, which is our automation platform where we're able to develop stem cell technology at scale. And this also includes our diverse iPSC bank, which I'll talk a little bit more about. One major piece of our NYSCF Global Stem Cell Array is our iPSC generation. We're able to have a fully automated iPSC reprogramming process that happens in high throughput. And for this particular collaboration with the National Eye Institute, we received whole blood samples from ARIDS 2 AMD patients. We were able to isolate PBMCs from whole blood and then put them through the automated process. So first we do reprogramming, which for blood cells, we reprogram with Sendai virus. We then put the reprogrammed iPSCs through a monoclonalization step to ensure that we're able to eliminate the presence of Sendai virus that might be sticking around from the reprogramming. We then move them into automated expansion, where we're able to generate stocks of these iPSCs and prep them for our QC. And then we have a full and comprehensive quality control panel that I will talk a little bit about in the next slide. Our global stem cell array is a fully automated and highly reproducible stem cell production process. And we first published about this in Nature Methods in 2015. So if anyone wants to check out a little bit more about that derivation platform, you can take a look at that paper. So I wanted to talk a little bit about the quality control that we do on all of our iPSCs on the global stem cell array. First, we have a viability test. So we're just checking to make sure that the actual cells that we distribute to anyone who requests them are able to recover well. We also do a sterility and mycoplasma test. We do our karyotype through the Illumina Infinium Global Screening Array, which is a genotyping chip. We have we classified as a normal karyotype if we are not able to find any autosomal copy number variations that are greater than 2.5 megabases, unless there's something that's known that's inherent in the patient already. We also do an identity test using the Fluidigm SNP trace panel, where we're able to make sure that the originating parent cell line, in this case PBMCs, matches the final iPSC that we're actually distributing. We also do a pluripotency expression profile. So we use the nanostring technology to take a look at pluripotency expression markers. Also check to make sure that iPSCs do not have any early differentiation markers. We also, at that same time point, check to make sure that there is not any Sendai virus sticking around from the reprogramming process. And then we also do a spontaneous embryoid body formation. And so then we'll take a look, again, using the nanostring at the differentiation capacity, and we use the scorecard analysis originating from Alex Meisner's lab, and we'll report the score for each of the 3 germ layers on a certificate of analysis that we generate. And each iPSC line that we generate does have a certificate of analysis, and so we'll provide that with each line that's distributed. To be able to obtain these AMD iPSCs, you can go to NYSCF.org/repository. The lines that were derived through this collaboration are, as I mentioned, all blood cells from ARIDs to patients, and there is clinical data available through the NEI Data Commons, which Kerry is going to talk about in a few moments. Our current website lists, our website lists the currently available iPSCs, and as Kapil kind of presented the table before, each line has the genotype for 8 AMD risk factors on the PDF that's up on our website now. We currently have 65 AMD patient iPSCs that are available, and each patient has 2 clones that are fully QC'd available. And there are 8 additional iPSCs, again, with 2 clones each, that are finishing up the reprogramming process. And so they'll be posted and the PDF will be updated in the next few months. If you'd like to request any of the iPSCs, you can email repository@nyscf.org. Please do include which iPSCs you'd like to obtain. On the PDF, there are 2 different columns with 2 different IDs, the NYSCF ID or the NEI ARIDS 2 ID. You can include either ID in the email of which lines you would like to obtain. Please just let us know if you would like to obtain 1 or 2 clones of each patient. Also include a brief research plan, which can just be a few sentences on how you plan to use the lines. Also the contact information for your institute's tech transfer office and the shipping address for where you would want to receive the frozen iPSCs. When we receive a request, we'll review it. If you have additional questions, we might go back and forth a little bit to provide some more information, and then we'll provide a draft MTA for your review and your institution's review. I did want to mention also that we have additional iPSCs that are available through our repository, including healthy controls, other diseases, a diversity panel. So if you have a need for additional iPSCs outside of just the AMD cohort, please do let us know. We're happy to provide you with more information. The fees for the AMD iPSCs are for academic groups, $450 per vial. If you do get both clones per patient, then it's just double the price it's $900 for 2 vials. The industry price is $950 for 1 vial or $1,900 for 2. And then the shipping costs are different depending if it's domestic or international, but for domestic, that's typically between $75 and $125, depending if you're East Coast or West Coast. We do ship on FedEx or via FedEx on dry ice and priority overnight. For international shipments, we like to use Cryoport to help with the logistics. They can help us with customs forms and specific country specifications. We use them as well, so we can use the LN2 dry shippers so that we can make sure the cells stay at cold temperature if they get held up for a few days in customs. And we'll usually get a custom quote depending on the country location once you make your request. And I also wanted to mention that these iPSCs are partially subsidized, the costs that I'm presenting here, by the NEI already. So when we do distribute our iPSCs, we provide an SOP for thawing, expansion, and cryopreservation. On our global stem cell array platform, we culture the cells in a custom media that is through Thermo Fisher. It's very similar to StemFlex. And so on our SOP, we recommend thawing into StemFlex or Geltrex and then transitioning to your preferred culture system. However, if you are familiar with some of the RPE protocols, often they start in E8. And so we have tested that you can thaw directly into E8. We definitely recommend using Cloner by Stem Cell Technologies for optimal cell recovery if you choose to thaw directly into E8. And we're happy to provide some phone call consultation before you start thawing the lines if you have any specific questions. And then obviously, as Kapil had mentioned before, these iPSCs can be differentiated into RPEs for disease modeling. And if you have questions about the differentiation protocols, you can go to the NEI AMD Integrative Biology website for more information. If you have any other questions, again, feel free to email that repository@nyscf.org email or check out our website at nyscf.org/repository. That website does have the currently available PDF with all the iPSCs listed. And lastly, I just wanted to thank everyone at NYSCF who's been involved, our NYSCF leadership team, Susan Solomon, Jeff Wallerstein, Reka Ayerick, Jeff McGrain, and Corvus Richardson, as well as our NYSCF scientific leadership, Rick Monsmo, Scott Noggle, and Daniel Paul. Definitely want to thank the rest of my project management team, Chris Hunter and Connor McKnight, who handle all of the logistics when we receive cells that come in and register the samples, to tracking the progress through our platform, and then actually distributing the iPSCs through the repository. And a huge thank you to everyone on our stem cell derivation group through the Global Stem Cell Array, John Cerrone, Camille Fulmore, Jenna Hall, Dylan Hudson, Dorota Moroziowicz, and Katie Reggio, among other lab members at NYSCF that have been with us the past couple of years working on this collaboration. They have done an enormous effort to generate all of these iPSCs and ensure that they have high standard QC completed. So thank you so much, and happy to answer questions in the Q&A portion after Kerry presents.
Thanks, Lauren. A lot of useful information. Lastly, we're going to hear from Kerry Goetz, the Associate Director of NEI's new Office of Data Science and Health Informatics. She will discuss how to obtain corresponding clinical data through the NEI Data Commons and give an overview of the data portal and other useful information such as what data is available and how to request access to it. With that, Kerry is loading up her slides and I'll let her talk about it.
Okay, thank you, Steve. I just want to confirm that I have the right screen share. All right, well, I'm going to go with it. Yes, thank you, Steve, for the introduction. For those who don't know me,
you've just got to switch your display setting, actually.
Okay, that's what I thought.
Thank you.
Okay, how about now? Thank you. Yes, so I'm Kerry Goetz. I am leading a new office at the NEI that is handling actually data management, data sharing, data science, and health informatics topics. And I am going to tell you a little bit about how to get access to data within the NEI Data Commons for the AMD Integrative Biology Initiative. And then I'm going to do a little bit of a demo as well, and then some more details about the application process. All right, so again, just be summarizing some of the data that has already been discussed, going over the portal and then the details. All right, so as mentioned, there are actually 64, I think is what Lauren said. So my details are a little slow, but 62 lines are what I'm gonna have details on for the demonstration today. Those 62 iPSC lines correspond to patients from the AREDS2 clinical trial, which recruited 4,203 participants. I've included the website here. It's also in the chat, and it will be in the slides for you. But the portal link is iPSC at .nei.nih.gov. Right. So for the iPSC lines, it represents 32 males and 30 females. They're all non-Hispanic, Latino, white participants between the ages of 65 and over 90. The graph here shows the distribution of the age at which the participants donated their samples to create the lines. There's a little bit of a bump at 90, and that's for privacy's sake. Anyone who was older than 90, their data has been changed to indicate they were 90. And this is a breakdown of the alleles that Kapil discussed. So you can see that some patients have, or a majority of patients have some alleles, such as the CFB L9H, TT allele, and then there's others that are more rare in this cohort. Okay, I'm going to do a demonstration now with videos. I'm going to go ahead and turn my video off momentarily just to make sure that I don't eat up all the bandwidth here and you can pay attention to what I'm presenting. But I'll come back on as soon as this is finished. All right, so first I'm going to show you how to find what data is available in the data commons for the AMD IBI. So when you go to our website and click on data dictionary, you're going to go ahead and select eye diseases. This data dictionary shows all the forms across all NIH applications that use this portal. But you can type in AREDS2 and get a result that shows all of the forms that are currently available. There's 17 right now, but we're continuing to add more over the next few weeks and as time goes on. And you can get a closer look at the data within each form by clicking on the name. All right, so each form has a GUID or global unique identifier and the subject identification number, which is the AREDS2 ID. And those can be linked to the iPSC lines. You can click on each specific data element and look at the short description for that, and then the permissible values as well. And I'll show you specifically for each of the elements, you can just click on it. So if we're going to look at this element ENZY5X, it corresponds to the highest enzyme level greater than 5x upper limit of normal for patients that had a myocardial infarction within 30 days after a bypass, and whether or not their enzyme level was greater than 5 times the upper normal limit. So a lot of detail contained in those data elements. And then the permissible value codes, so indicating either, yes, the patient had the 5x upper limit of normal, or no, they did not. Okay, so now I will show you a demonstration inside of the NEI Bricks Data Commons. Right. So again, from our website, once you're allowed access, you would click on Login and then go to the Query tool. So I'll pause it here for just a second, just to say that the NEI Data Commons contains several projects that I'm not discussing here, but want to just put a quick plug out for. iGene, which is rare inherited eye diseases, and a LASIK quality of life clinical trial data are also available. And we anticipate adding more clinical trials to this portal for access as they become available. The benefit of this is that you can combine different projects. So if there's studies that make sense to be linked, this portal allows a user to do that with the right user requests. Okay, so once you've decided you want to look at the AREDS2 data as well as the AMD Integrative Biology data, you would select those, add them to your data cart. Within the portal, you can drag and drop up to 5 forms to combine them. Doesn't require a whole lot of coding experience, pretty user-friendly. And then we can further refine the data selection through filters. All right, so for example, I'm going to go ahead and refine my data on the presence of those 5x enzyme levels. Within the portal, you can look at the elements of that data element again. So I might not remember what's the code for yes or no. I want to look back and then select that. So I'm going to select only participants with that upper enzyme level. And you can see I have a query box open, so you can look at the system logic and further refine. And now I want to only select the oldest participants who donated a sample. You can see that I can use the filters and Boolean functions to do that. Okay, so because I can't show participant level AREDS2 data in the public, it's controlled access sample, I'm going to go ahead and clear this deck here and just give you a look at the iPSC data. So you can actually get a sense of what everything looks like when you're doing it live. Okay, so once again, I'm adding the forms that I want to join. So this is actual biomarker data plus some other data that we have on the actual cell collection. And now I want to just find participants who have a specific CFB gene allele. You can see here I can do inclusive or exact on the filter. Exact is useful, especially for gene names that might be within a family. And then now I want to further select on the L95 or L9H allele. And you can see the Boolean function. So I want CFB and the allele. And now I just want a specific variant. And I can go ahead and find that. Then I can add data from the second form and look for just patients who donated at a younger age. So say I just want people under 70. And then go ahead and run that query. And you can see that the database returns my relevant cohort here. And you can see in the table, you can find the NYSCF IDs to make the request to NYSCF to obtain those. You can see the ARIDS2 IDs, which again can be used to make those requests. And then the details about the participants. Within the data, the data table, you can apply filters, you can hide columns, you can customize this view however you'd like. And you can also save your query. So if you want to run this later when there's more data added, you can continue to run that. You can share it with collaborators from within this system as well. And you can also just download this data to your computer using that, the download feature. Okay. All right, and then so one last pseudo demo for you. So this is the requesting access demo. So it's pretty simple. This one should be quick. To request access, again, you just go to the iPSC website, click login. You're going to request a new account. And within the account, you're going to go ahead and load the required documents that you need to make a request. The request go to an access committee for review. And I'll give you a list of what documents are required for review here in just a second. So you simply upload all those documents and then complete a username and password request and fill in your contact information. And then the NEI Office of Data Science gets an email and we can route that for review. All right. The data access request needs to include a bio sketch or CV or resume. The proposal itself should have a title and describe the information sought. This is really important because we review each one of these just to make sure that your request is actually, the data within the Data Commons suits your request. We'll need a description of the project and a timeline. Also data management and sharing plan. So the main objective of the AMD IBI is to create a robust knowledge base. And as such, we will require a data sharing and data management and sharing plan. Essentially, we want to know what data you're going to create or collect and how you'll share that back to create this resource. And then if you need IRB review, we'll ask for that documentation as well. All of the details that are described here are in that on the website on the how-to column. And then once you're approved, we'll create a data user agreement, which includes statements saying that you'll report annually to us. And then that data will be included in the NEI Bricks portal whenever possible. So again, people can join those different data sets to create a more robust resource for the rest of the community. Okay, and with that, I will just leave it open for questions, and my office is happy to take any additional questions or have one-on-one conversations as you'd like. Okay, and I will flip it back over to Steve. Thank you.
Thanks so much, Kerry. That was great. Now we're going to go into our Q&A portion of the webinar. We have a little over 15 minutes. So to kick us off, I see one question in the Q&A, and I'm going to ask Kapil to answer it. Are there any plans to generate isogenic controls for the NEI AREDS2 iPSC lines?
Yes, the short answer is yes. If you remember the table I showed you about the 11 lines, all of them had the CFH risk allele, and there are two risk alleles were present in all the lines. So we are systematically converting them all in both the CFH risk alleles into non-risk. So you'll have isogenic lines available for those. As it stands right now, we are hoping that these lines will be ready towards the end of this year, maybe by fall or slightly later than that. And they would be hopefully distributed through the NYSCF portal as well.
Thanks, Kapil. We've had a couple general questions about if this presentation will be available later. And so I just wanted to let folks know that this is being recorded and that it will be posted on New York Stem Cell's website. And we'll be promoting that through our social media channels, and we'll let you know hopefully in the next few days when the content is available. Any other questions? I'll pause. I know in general, there might be some questions of, what's the extent of this collection? How big is it going to grow? Lauren, you want to take that on?
Yeah, so as I mentioned in the presentation, there are 65 currently available lines, and then we have 8 more that are just finishing up. So in total, it will be 73 iPSCs, and then of course, each of those have 2 clones each.
Thanks. I see a question that just popped up. How many passages for these iPS RPE cells? Passages,
So
I guess.
If we mean just the iPSCs, they're provided somewhere around passage 10, and we do include the passage number on the certificate of analysis that we provide when we distribute the line.
And if I may add to Genia's question, for RP cells, the way we make it, we don't passage them at all. We use them as a primary passage 0 cells.
Great. Any more questions? I know we've talked about the science. Any more about the NEI Data Commons and the data associated with these cells? We got some coming in. Someone asked generally, what are the risk allele frequencies in the general population?
Well, it depends. I would say it depends on a risk allele. And this is going back to the GWAS studies that led to discoveries of these risk alleles. And obviously, the distribution of these specific risk alleles was higher in the AMD population. That's how they were discussed. That's how they were discovered.
Thanks. We have a question asking about protocols for photoreceptors or choroidal endothelial cells. Will they be available?
You want me to take that?
Yeah,
John, for endothelial cells, there are published protocols. I can send you publications that we are following in my lab. These are not the protocols that we have developed, but anyone who's interested, we're using published protocol for both endothelial cells and photoreceptors.
We have a couple generic questions about, how long is it going to be for translation of site-saving therapies, basically, to repair the retina? I know, Kapil, you have an ongoing trial. You want to give that a stab?
Sure. Yeah. So that's kind of independent of this research effort. That's more in the clinical realm, where we have NEI has an ongoing clinical trial, phase 1/2A for autologous iPS-derived RP patch for AMD patients. If you're interested in that trial, perhaps we can share the link with you here. Rick, if you have access to the link for the trial, do you mind pasting it in the chat?
We have someone asking about the risk alleles that were selected, which reference publications were used?
I would say there were a number of publications from the literature that were, all of those were collated together to come up with a list of highest risk alleles. And of course, we were limited by the access to ARIDS2 patients that we wanted to use for this study. And in fact, NEI had a panel of experts in the AMD field come together and kind of prioritize those risk alleles and just suggest the highest possible risk alleles that could be used, that were going to be used to make this iPS cell lines. If you're interested in the publications, perhaps we can post them on the website later.
Yeah, there's.
I'll just add to that, Kapil. I know we did post a couple references on the iPSC.nih site, and we can certainly add more there.
Thank you, Kerry. That's wonderful. Yeah.
Yeah, in the chat, we've been putting links to different web pages for more information about this program and related activities. So please take a look in the chat. There's a lot of useful links. Any other questions? I guess, Lauren, in terms of you referred to some support that you provide. Can you go into a little more detail on what kind of support you might offer for people that obtain these lines?
Sure. So we, as I mentioned, do provide an SOP for thawing and expansion and cryopreservation of iPSCs. So oftentimes when someone might request lines, if you're newer to culturing iPSCs, we are happy to jump on a Zoom and really review the protocol, answer any specific questions. And then again, if you do have any issues thawing the lines, we're happy to take a look at pictures, help walk through what you might need if you need to make any adjustments, or just make sure there weren't any steps that were missed or anything like that. We're very happy to get on as many calls as you need to help make sure you have success in being able to grow these cells out.
That's wonderful. Thanks so much. Any other questions? If not, I just want to let people know that the Office of Regenerative Medicine at NEI is open for questions as well. We're happy to explain, this program in broad terms and refer you to experts that may have the expertise that you're looking for to help you differentiate cells or to use these cells. It's really a wonderful resource that I hope folks here have come to appreciate through this webinar. And again, if you have any questions, please reach out and we'll be happy to answer them. All right, I just see a lot of thanks and other comments in the chat and Q&A. With that, I'll conclude our presentation for today. Thanks everyone for your attention and your questions, and everybody take care.
Thank you, everyone. Thanks to the team.
Thank
Take
you.
care.